start the recording at your convenience. There will be a time for question and answers after the presentation. So please either hold your questions until then, or if you want to throw them in the chat, we can scroll through at the end and address them then, unless Dr. Thomson and Dr. Fischer prefer to answer them as you go. That will be up to how you guys want to handle it. Okay, I'd like to introduce our speakers for today. Dr. Joshua Thomson is an assistant professor and interim director of research administration at the University of Detroit Mercy School of Dentistry. He teaches microbiology and immunology to dental and dental hygiene students and residents, with a specific emphasis on infectious diseases and infection control. Dr. Thomson received his Bachelor of Science in Biology from Detroit Mercy and his Ph.D. in Immunology and Microbiology from Wayne State University School of Medicine. His main research focus is directed towards the use of natural compounds and bacteriophage as therapeutics against dental pathogens, as well as defining microbiological characteristics of dental materials. His continued research interests include describing mechanisms of serum resistance in the human bacterial pathogen Yersinia pestis. I was interested to see that. I once had a patient years ago when I was still practicing nursing with bubonic plague, and it's not something we had seen at the time at all. And our other presenter is Dr. David Fischer. He also is an assistant professor at the University of Detroit Mercy School of Dentistry. Dr. Fischer teaches immunology and nutrition to dental hygiene undergraduate and graduate dental students. Dr. Fischer has a Bachelor of Science degree in cellular and molecular biology from the University of Michigan and a Ph.D. in immunology and microbiology from Wayne State School of Medicine. He has conducted postdoc research at the Ohio State University's Agricultural Research and Development Center. And his current areas of dental research include the roles of human herpes viruses in periodontal disease, antiviral effects of propolis and essential oils, and the interplay between nutritional status and the adaptive immune response. He has previously conducted research on influenza and rotavirus vaccines. So welcome, Dr. Thomson and Dr. Fischer.
Thank you very much for the introduction. It's a pleasure to be here. So I'm gonna share my screen now. Hopefully you can see a slideshow. Does that seem right? Awesome. I'm going to try and give remote control to Dr. Fischer. So we'll kind of go back and forth with this. And can you request it, Dave? Perfect. Cool. So again, I'd like to just thank you for the invitation. This is awesome to be here and doing this. Like I said, I'm from home, and so my kids are running around doing something right now. And so you might see them come into view or me lean over and yell at them. But again, thanks. We've been doing a lot of presentations on vaccines and COVID-19 over the past 12 months, it seems. And anybody that we can provide information to or answer questions for, it's something I truly enjoy, and I think Dave as well. So with that, my attempt at a fun title was Viruses, Vaccines, and Variants, Oh My, What You Need to Know About SARS-CoV-2 and Why. So hopefully we will hit on everything that you've been wondering about. Learning objectives here, I usually don't go through them. I'd rather just get into kind of going through the talk here. But I wanted to take a step back and just talk about what is a virus first. So if we're defining it, a virus is an infectious obligate intracellular parasite that's comprised of genetic material, DNA, RNA, surrounded by a protein coat and or a membrane. So here, the classic T4 bacteriophage that looks like the lunar landing craft. This is from one of our dental students in an art competition this past May. I created all these on some Adobe program that I don't know how to use, but he created these and gave me permission to use them in my lectures because I thought they were awesome. But here we have just some different other examples of human viruses. And so they're capable of directing their own reproduction, and they spread by taking over the machinery inside of a host cell that it invades. So they got to get into a host cell. They take over that cell to produce more of them. And in classes, I've had students just kind of try and come up with different ways to little analogies or anything. And I like to refer to them as infectious recipes. So if you can deliver that recipe to the cell, that cell will cook it up and make a new virus, whether they want to or not, usually. And then one thing that I like to just put out there is that they don't independently fulfill the characteristics of life that we think of as alive or something that's alive. So we don't really refer to viruses as alive, or I should do my best to try not to. We refer to them as infectious or not. Yes, we are dinner. I like it. So when we talk about viruses and, you know, alive or not alive, really anytime that's being said is infectious or not infectious. So can they infect a cell and replicate? I had a dental hygiene student last year refer to them as little bags of DNA or RNA. I kind of liked that as well. Just some, this is a really cool book, an illustrated guide to 100 microbes. I found this, 12 months ago, and it has a bunch of electron micrographs of viruses that are pseudo-colored like this. And just to give you an idea, there's plant viruses taking various different shapes. We have these giant viruses. These are viruses that infect amoeba. So we have the Mimi viruses, the pithoviruses, the Pandora viruses. These are kind of really new findings once we started doing sequencing rather than just filtering these little viruses away from bacteria. We're finding things that are actually bigger than bacteria. And then some of the human viruses, I chose these because I think they're kind of my favorite. We have the bullet shape of the rabies virus, HIV with its mature virus capsid in here and a non-mature virus over here. And then variola virus is the causative agent of smallpox. And then human papillomavirus 16, a virus that's associated with causing oropharyngeal, cervical cancers. And so just some of these variations in what they can look like. But if we look at all the viruses, what are they? What are the parts of viruses? And so here we have two different kinds, a non-enveloped virus and an enveloped virus. If we use the term virion, anytime we use that, we're really referring to an infectious virus particle. Infectious virus would be a virion. A particle would be something that, if we say just a virus particle or a particle, referring to something we don't really know if it's infectious or not. We just know that it's kind of there. We expect it to be there. A lot of times, all three of these will be used interchangeably without really any regard to the definition. The genome is going to be that recipe to make new viruses. So when it's just like our chromosome is DNA, has genes that turn into protein, a virus will have those same things, DNA or RNA though. So they can encode their genome on an RNA nucleic acid, whereas we would just have ours encoded on DNA in our chromosomes. And they have a protective protein shell called a capsid that will cover this nucleic acid. And in a non-enveloped virus, that's the outside. In an enveloped virus, it's surrounded by a lipid bilayer, just like our cells are. So it steals a part of our cells as they are released from an infected cell. And on the surface, they're going to have glycoprotein spikes, so these spike proteins that stick out, kind of like they look like aliens to me, alien craft. They're going to stick out, and those are going to be what are really important for a virus to bind to a cell that it's going to infect. And so you've heard lots about spike protein. Well, basically every virus has some version of a spike protein. And then one other distinction, just kind of starting off, that I wanted to make is the difference between infection and disease. So when we talk about an infection, a pathogen or some sort of agent that can cause disease, an infection is going to be a colonization or replication. If we're talking about viruses, a virus gets into a cell, replicates within it. That is infection. So an example, the virus SARS-CoV-2 infects lung epithelial cells. That is an infection. We are infected with lots of viruses all the time without even knowing it. Many of us here are positive for different herpes viruses like Epstein-Barr virus, cytomegalovirus, herpes simplex virus 1, varicella zoster virus, the causative agent of shingles and chickenpox. We're infected with all these lifelong without ever knowing it. And that's just a part of the amount of viruses that we're infected by. And then on the other side, disease is when one of these infections does something to our body, prevents it from functioning normally, and leads to signs and symptoms. So usually when clinical disease is something where you have outward showing of the infection that's going on. So an example here, COVID-19, that's the disease. So SARS-CoV-2 infects lung epithelial cells, leading to the signs and symptoms, maybe a cough, fever, all of those symptoms that we talk about. Many viral infections result in no symptoms, mild symptoms, or they can go to severe symptoms or death. And I thought this was another art in our microbiology art contest by Jay. And I thought it was a good representation of the virus and the disease in one picture, where we have the SARS-CoV-2 virus along with one lung and then one more diseased lung over on the other side. Dr. Fischer.
All right, so a couple terminology things here, following off the last slide there. So you probably hear, the term COVID-19 thrown around. You probably heard SARS-CoV-2 thrown around too. What exactly do they mean? So the virus itself is SARS-CoV-2. So the SARS stands for severe acute respiratory syndrome. The CoV part is coronavirus, and the 2 is because it's related to but different from the coronavirus outbreak that caused the 2003 SARS outbreak. So the disease is COVID-19. The SARS-CoV-2 infection can lead to COVID-19 disease. So the nomenclature here, the CO from corona, VI from virus, D for disease, and 19 because it first emerged in 2019. So there's no COVID 1 through 18, it's COVID-19. And I've done a lot of the contact tracing for the dental school here, and it's really amazing how different the disease is in varying people. You had some people come in and say, hey, I thought I just had allergies, but I went to get tested and I came back positive. They had nothing else other than maybe a little sore throat or a tickle in their nose. Other people, it started off with a headache. They figure, all right, I'm a stressed out dental student. I'm tired. I haven't been sleeping right. So maybe that's a headache from that. But then, some people it doesn't go beyond that, but others 2 or 3 days in, some lose taste and smell. Others have a fever. Some develop respiratory symptoms. Some don't. In the serious cases, some people are out for a while. Others, other people end up in the hospital. And unfortunately, we've had a lot of people lose their lives. But these collection of symptoms all fall under the category of the COVID-19 disease.
So human coronaviruses, or HCOVs, so if we're trying to just categorize them, they're in the Coronaviridae family. They get the name was originally given for the crown. If you look at an electron micrograph here, it's not the greatest of pictures, but when you look at it from a cross section, you see all these spikes coming off that sort of look like the corona of the sun or a crown sitting on a head if you're looking down from that. At least that was my interpretation of how it looked like a crown. It took me forever to even try, and I didn't see crown. I don't know. So the genome is an RNA genome. It's single-stranded, and coronaviruses have the largest genomes of known RNA viruses. Usually RNA viruses are a little bit smaller. And so in order for this to make and replicate their genomes, all of the coronaviruses, they have an RNA, they have proofreading enzymes. So if they're making mistakes in the RNA, usually they'll get fixed. Or because if they did not have that, the mutation rate would be so high that they would be losing really important enzymes and gene products that they need for the replication of the virus. Everybody, I'm sure, has seen the classic CDC SARS-CoV-2, showing that it is an enveloped virus. They have these spike proteins on the outside. So it does have that lipid bilayer membrane on the outside. So generally, they're pretty susceptible to drying out or desiccation and detergents. And so, hand sanitizer works to effectively eliminate infectiousness of the virus. Asymptomatic infections are extremely common with coronaviruses. Thus the problem we have now, because there are people that don't exhibit signs and symptoms of COVID-19. They're asymptomatically infected, but they can still transmit the virus to other people. We have common cold coronaviruses. There's 4 of them that we know of, and they cause about 15% of seasonal colds, alongside the rhinoviruses that also cause them. And they jumped to humans from other animal hosts at some point in our history. Dr. Fischer knows a lot about these in the lab that he was at the Ohio State University, and coronaviruses can infect animals and humans. There are a lot of animal pathogens, agricultural pathogens. We know they can infect bats, cats, rabbits, mice. I can just read all these, and we probably heard a bit of the mink issue with these viruses going through, with SARS-CoV-2 going through minks. But really, the big thing that we're worried about is, well, now we all are worried about it, is that severe infections with these can occur when they jump species. So when they go directly from an animal to a human, where humans have never seen them immunologically before.
Let me take this one.
Yep.
All right, and so keep the spike protein in mind. I don't know why it's coloring like that, but the spike protein is very important. That's the first thing the immune system sees. And also remember, as Dr. Thomson said, the virus can't replicate on its own. So like a bacteria can grow, if it has the right nutrients, can grow on just about any surface. If you cough or sneeze out a virus and it lands on the table, it's not going to be able to reproduce itself there. It needs to get into our cells and use our cellular machinery for it to be able to grow. And so it uses the spike protein to grab on to the molecule ACE2 that's on the surface of a subset of our cells. And so after it binds, the virus gets into the cell, it releases its RNA genome, and then it uses our ribosomes. So it hijacks our cellular machinery to make new viral proteins. So after it makes new viral proteins and it replicates its nucleic acid, it can assemble, and then new viruses can bud out and go on and spread to the next cell. Now, if a person's immune system catches it, or the virus kills a person, it needs to go on out somewhere and find new cells. So it goes and tries to jump to the next host.
So I think what happens is I try to move it at the same time you do, and apparently it like clicks and highlights.
Okay.
But I wanted to just mention on this a fairly busy map here, but SARS-CoV-2, because of its spike protein, it has 2 different distinct ways to get into a cell. And I think it's important when we talk about why some different therapeutics just don't work. So one way over here is what Dr. Fischer mentioned on the previous slide, where it can bind to the ACE2 receptor that triggers what's called endocytosis. So it gets taken up in this little membrane, and then gets the spike protein gets cleaved by some proteases in the endosome, and then it's able to fuse the membrane in there and be released into the cell. The other mechanism is where that proteolysis of the spike protein happens at the surface. So temporis 2 and some other proteases outside of the cell can cleave that spike protein and get it ready in its pre-fusion state right there, ready to fuse at the membrane. And so it doesn't need this endosome. And so like, hydroxychloroquine was one, and Dr. Fischer will talk about that in a little bit. It plays a role in this pathway, but there's this other distinct pathway that with SARS-CoV-2 almost gets used more frequently. There's some data showing that. And so hydroxychloroquine wouldn't work to stop that other pathway over here. I'm just gonna mute before you take over.
All right. And So it's important to remember that when you're sick, you're feeling the combined damage of what the virus or bacteria or whatever is doing to you. And then also, when you turn the immune response on, it's a very powerful process. It takes a lot of energy, and it's also a very damaging process. So, if you kind of think of warfare going on, there's going to be collateral damage. So how does the virus get in and cause disease? So if you never encounter someone with SARS-CoV-2 or you never encounter the aerosols that they've coughed out or breathed out, you'll never get the virus. Now, say you're without a mask, you're in close contact with someone who has it, they breathe it out, you can inhale those particles, those droplets. After that, it gets down deep into the person's lungs and it binds that target ACE2 receptor. So then it starts getting into the epithelial cells in the airway, starts replicating, it damages, destroys some of those cells. After it's used up all the resources, virus goes on to the next cell, and at the same time, you're breathing it out or coughing it out. And so a lot of people, the disease doesn't really progress too far beyond this. In other, it's milder symptoms, it's cleared out pretty easily. In other people, the virus can get a little bit further along. So it reaches alveoli and type 1 and 2 pneumocytes, so cells that are heavily involved in the oxygen exchange process. And so when we have an infection, first we trigger our innate immune system. So this is a very inflammatory, non-specific response. It's good at taking care of a lot of infections, but it causes a lot of damage too while you're building up your antibodies and your T cells and your B cells. And so an inflammatory response kicks off. So when you have inflammation, you start bringing additional cells of the immune system. Now those cells come in and see that there's an infection happening too, and they start kicking out all these inflammatory compounds and cytokines too. So as you can imagine, you're going to have kind of a feedback loop here, and the inflammation will really build upon itself. And in severe cases, the virus can leak into circulation and start affecting multiple organs. So this is what leads to like heart failure in some people. Also, when you're having the inflammatory reactions in the lungs, you're getting a lot of fluid secretion as well. So the virus is damaging and infecting the cells that are responsible for the airway, for the oxygen exchange and the lung function. Then you start throwing over this layer of fluid, and that's when the severe pneumonia can happen. The person might have to end up on the ventilator or need supplemental oxygen. I'll take this one.
So these are not exactly up to date right now, but I think they gave a good idea of just where we're sitting with COVID-19 infection. So this is looking at symptomatic illnesses and hospitalizations in the US, February to December 2020. So last updated January 15th here, but this is part of the reason why it's such a big issue. 1 in 1.9 COVID-19 hospitalizations were reported. So this is still under-reporting. 1 out of 4.2 symptomatic illnesses were reported. 1 in 4.6 infections were reported. And new data is coming out as we get better with testing previous infections. We get a better handle on that. We're starting to find out that in certain subsets of age groups, it's even more highly underreported, especially in children. We're looking at millions of infections, millions of hospitalizations, millions of symptomatic illness, because everybody is susceptible.
Yeah, and a lot of people are saying, oh, the numbers are underreported. We might actually have more protection than we think. Yeah, we have some more protection out there. There's probably more people immune than we know it, but it's still nowhere close to being enough to provide complete protection or herd immunity in any of the regions. But I mean, and this is kind of a reflection of how lousy of a job we did with testing, especially in those last spring and early summer.
When nobody could get testing, they're just like, yeah, you probably have it, but you're younger, you're not at needing the hospital, so we're not going to worry about it. This was one of the studies I was referring to, and so it's talking about seroprevalence, so the amount of people that have antibodies. So if you have antibodies against SARS-CoV-2, and that's an indication that you had a previous infection. Now, depending on what antibodies you're looking at, it might be an indication of a vaccination. But here they were looking at persons aged less than 18 years in Mississippi from May to September 2020. So it's a retrospective study, and it's a convenient sample. So it's not the perfect representation, but they had residual blood specimens from a blood bank in these children. They tested 1,603 for anti-SARS-CoV-2 antibodies, and 175 of them came back and tested positive. They had a pretty good representation, and then they were able to extrapolate that data out for the actual population in Mississippi. What they found was that in this study, non-Hispanic Black and Hispanic young persons were 2.4 and 4.3 times respectively the rate among non-Hispanic white persons. We've been seeing that all the time with this, that there are ethnic disparities. After adjustment for race and ethnicity, seroprevalence increased from 2.5% to 16.3% in September. So that's the number of new infections. And this was just mind-boggling to me. When they took those numbers and took them out to the entire state population, this 16.3% number in September 2020 suggests that 113,000, almost 114,000 of the almost 700,000 young persons in Mississippi might have been infected by the end of September 2020. Similar to the rates in age 18 to 49-year-old. But during this time, there were only 9,000 confirmed and probable cases in those age groups. That is 13 times, up to 13 times the number reported that could have been infected. So as we were debating about, can young people get it and transmit it? Yes, and yes, we know this about viruses and respiratory infections. But why are we not seeing it? Children are under-tested. Partially because they're less likely to have as high symptomatic cases. And we know now that there's a shorter period, a time period of symptoms for children, as well as positivity by the PCR test, the RT-PCR test that we do. So even if you were testing them, there is a smaller window where it would be positive for children. But then also, anybody with children knows, is it worth taking them somewhere? Sometimes you could argue it's not worth it, but here I think it should be.
All right, and we're learning a bit more about some of the longer term consequences of SARS-CoV-2 infection and COVID disease. So the technical terms post-acute sequelae, you may hear it referred to as long COVID or the COVID long haulers. And some of the most common symptoms are loss of taste and smell. We've talked to some people who have lost their taste and smell for up to 3 months. And, that actually has a pretty big impact on the quality of life. One of my friends lost it over Christmas, and he said that really put a damper on the holidays. But, fatigue, shortness of breath, people can get very winded doing what should be a relatively easy activity for their age and otherwise, good health. Coughs, joint pain, and chest pain can last. My wife's doctor says that she's been dealing with brain fog. She forgets stuff. She has to take a lot more notes, and it's lasted for a few months after. Other people end up with depression, various aches and pains, headache, heart palpitation. And then also in the more severe cases, these aren't as common, but, as I said, the virus can spread to multiple organ systems in serious cases. We've seen things like myocarditis and some, high-profile athletes. Issues with the lungs, issues with the kidneys, a rash. Some people are losing hair in the months after. Again, sleep problems, smell and taste issues, concentration, memory problems, depression, anxiety, and mood changes. And there isn't a really good explanation for a lot of these. This is going to be an interesting area of research.
And that study at the bottom here, it was a pretty recent one where they did a web survey of almost 4,000 patients that had long COVID in 56 different countries. I wanted to highlight here, there is a female disparity, or maybe that's not the wrong word, or the right word, but it occurs more frequently in females, which to me speaks that, females generally have a higher antibody response to things. And so this might be antibody driven. but that's me just talking out loud. Some of that very infrequently were the people that had long COVID hospitalized. So this is mild or asymptomatic infection, and those are the ones that are more likely to have long COVID almost, it seems. 27% of them reported laboratory diagnosis. So there were plenty that were not actually diagnosed, but they had COVID. Nearly, I don't think I said that right. Anyway, nearly half could not work full-time 6 months after their infection. Fatigue, post-exertional malaise, so that was getting winded, feeling tired after doing something minimal, basically, and then cognitive dysfunction. Relapses would be triggered by exercise, mental activity, stress. When I hear that with mental activity, I think of concussions almost, where it can trigger the onset of, it's bad for recovery if you're thinking too early after having a concussion. So I like students going back to school after they have one, they're actually now being told to stay home more, so they're not thinking that early after a concussion. That kind of reminded me of that. Another new thing that we're finding more and more is that long COVID is happening in children that get it as well. So this preprint, preliminary evidence on long COVID in children. So it looked at children less than 19 with lab confirmed COVID-19. We had 129 with a mean age of 11. They classified them. 3 of them had multi-system inflammatory syndrome, which is pretty nasty. 2 had myocarditis, heart infection or heart inflammation. And then 53% of these children were reported to have 1 or more symptoms 4 months after their diagnosis. Insomnia, respiratory symptoms, nasal congestion, fatigue, difficulty concentrating. But within the young population, it was more prevalent in this particular study in symptomatic acute COVID-19 cases, but they did still see it in those that had asymptomatic. And anecdotally, I've heard a story from somebody on a podcast I listened to. There was a 16-year-old like track star in high school and got COVID and now can't do it 4 months later. Basically, the end of her high school career, just couldn't go back and track. And I don't think that was just a one-off story based on what I'm reading. And then looking at US pregnant women in SARS-CoV-2 infection, we know that there is increased risk of severe COVID and if you are pregnant when you get infected. And so we looked at, this was back in June, the study, and we have more data on it now, but there was an increased risk for hospitalization and ICU admissions for pregnant women that were infected. And Hispanic and non-Hispanic Black pregnant women appeared to be disproportionately affected during pregnancy as well. So we know that COVID is a risk for pregnant women if they get infected naturally while pregnant.
Yeah, and in general, pregnancy can lead to temporary immunosuppression as well. So that data makes some sense.
Because we see that with influenza as well.
All right. So again, since SARS-CoV-2 cannot live and replicate on its own, if we can break the chain of infection, that's really important in stopping the spread of the virus and stamping out this pandemic. Again, needs to rely on cellular machinery to grow and replicate. Now, if the initial person who's infected dies, or even if they're having an immune reaction and antibodies that now make the body inhospitable to the virus, but it needs to get out and get to the next person. So the next thing is it has to get out of the body. Usually that's through the mouth and nose, typical for most respiratory viruses. Then it has to get to the next person. Once it gets to the next person, just because you're exposed to a disease doesn't mean you're going to get it. The virus has to get into the body and then get to the target cells. And then finally, it has to get to those target cells and replicate. So if we can break any link in this chain, we can slow the spread of disease. So best way is distancing. You don't come into contact with someone who has the virus or their secretions or their droplets, you're not going to get the disease. So that's why we have these keep 6 feet apart all over the place. There's a lot of data supporting that 3 feet is a really crucial mark, and then that effect doubles when you get to 6 feet. Now, you can also block the virus from coming out of the infected person. Masks are very effective at doing so. So the person breathes those virus-laden droplets out. The mask should be able to grab onto them, and then it'll dry out in the fabric, and the virus will die there and not spread to the next person. Now you can also block the portal of entry. You can do this through good hand sanitization and cleanliness. So, you use the hand sanitizer, wash your hands. You can get some of that virus off of yourself, and then you won't touch your eyes, nose, and mouth. You mask up as well. That will catch some more of those viral particles from getting in. And if you have each person wearing a mask, that will help further reduce the risk of transmission. I see a question about, how about avoiding aerosol transmission? Yeah, I mean, the masks are very important in that. Air filtration, keeping the air moving, being in open spaces, that's all very important as well. But again, masking are really, really effective at blocking the chain of infection.
So I think just to add to that, I think what we know is that, so aerosol meaning remaining suspended in the air, not just being projectile and falling down, that is not the main route of transmission, and it is a possibility, but yeah, like Dr. Fischer mentioned, having good air control is important.
There's a lot of semantics involved here. It's like, when's it a droplet? When's it an aerosol? Is droplet transmission count as airborne transmission, or is it only aerosols, that count as airborne transmission? Those droplets, they're a lot heavier. They fall to the ground quicker. They have a lot of virus in them. The aerosols, yes, they can get farther. They can hang in the air for a longer period of time, but there's much less virus in there. So yes, the aerosols are smaller. They can get deeper down into the lungs easier, but there's less virus in them. So it really seems that the biggie is those droplets that, spread in close contact areas. Now, some viruses we have antiviral drugs for. HIV, if a person's on highly active antiretroviral therapy, they can knock down the viral loads in their body to the point where they're undetectable. And at that point, the person cannot transmit the disease through sex. We do not have any really effective antivirals at this time for SARS-CoV-2. Good news is we have vaccinations now, and the vaccinations can protect the susceptible host. So if the sick person exposes the vaccinated person should have antibodies that will block the virus from getting into their cells. So if we don't institute these methods, on average, one person with SARS-CoV-2 would infect somewhere between 2.5 and 5.7 people. We were seeing that early on in the pandemic, where we had that exponential spread.
So there's a question, how about 3-foot guidelines from the CDC for children in school? My wife is a high school teacher, and she goes, oh yeah, they're just now getting those guidelines. There's no way to have a class with 6 feet distancing unless you're going to split the school up, and that's just not going to be a thing. And so why does 3 feet guidelines work? Because they also, I think, because they're also instituting masking. And that, I think, is going to be controlling a lot of it more so than anything. But there are some other, in that study that I believe you're mentioning, there are some other nuances to that study that just weren't necessarily highlighted right off the bat with the types of schools that they were able to look at for that retrospective study.
So, one way we stop transmission is avoiding infected people. With some diseases, that's easy to do. Certain diseases, a person does not transmit the virus until after they already show signs and symptoms. Our biggest problem and the biggest issue we've had is that SARS-CoV-2 does not, or is highly contagious before the symptoms appear. So here on this chart here, it's days after symptom onset. So day 0 is when the person would start feeling ill. And this is the infectiousness profile on the y-axis here. So you can see the person is most contagious a little over 2 days before their symptoms actually appear, and they're most contagious about 16 hours or so before they start showing symptoms. So that person's probably walking around feeling fine, maybe not being on as high of a guard as they should be, and they spread the disease then. SARS-CoV-1, the original SARS back in 2003, we were able to stamp that pandemic out because it wasn't contagious until after symptoms appear. So if a person started coughing or having respiratory issues or spiked a fever, stick them in a room, keep them away from other people. You're able to control the spread there. But since we have seemingly healthy people walking around spreading this disease, it makes it really hard to contain. Also, you may have been asking where those 10-day guidelines from the CDC come from. Why can you return to work at 10 days out? Why don't we require testing for someone to come back to work or school? Well, the infectiousness declines to almost nothing after 7 days, and we throw in 3 days worth of buffer to add an extra layer of protection. But also, dead virus is going to remain in a person's system for a period of time, or abortive infection could happen as well. So even though nucleic acid could be detected, it doesn't mean a person's contagious. So, this is why we go to the time period rather than looking for multiple negative tests. Super spreaders, it's really interesting. It seems that about 20% of cases are responsible for about 80% of transmission.
So a question, thank you for the comment and great explanation. Appreciate that. Are there sources we non-experts might use to keep aware of these issues? Honestly, the CDC has been, I think, their website is for COVID-19. If you just Google CDC COVID-19, they have some really good flyers and stuff for promoting this information. But sometimes when it gets reported on, it isn't the best. Some people on Twitter have really good ways of describing it. And Dr. Fischer, you have any other comments about?
Well, yeah, CDC website, it's great. I mean, you can find all sorts of different scenarios. If you have to be a caretaker, if you yourself get sick, when should you get tested, what kind of test, and then it can get deeper in there for healthcare providers, workplace type things. So that's the place I would start. And then a lot of those will have reference studies as well.
And the CDC is under new management, and so is the country. And I've really liked how it's progressed in how they are giving out their information over the past 6-7 months. In the very beginning, it was a little bit more difficult.
All right. So again, the main way that the virus spreads is close contact in droplets. So close contact defined as being within 6 feet of someone for 15 minutes in a 24-hour period. So people were kind of playing games with the early definitions. Early on, close contact was defined as 15 consecutive minutes. So some schools were doing things like, oh, we could do 14 minutes on, then 1 minute off, and then start up again. No, that's not how it works. That's why they went to the cumulative time. And so these aerosols and droplets, it's a much more common route of spread than surface transmission because there's a much lower infectious dose. If you get this spray, and most of this is unseen. If you breathe that in, it gets down into your lungs, you're getting a very high infectious dose and can establish an infection very quickly.
It was hand drawn by me.
And so breathing, speech, sneezing, coughing, certain medical procedures, for us in the dental field, that's very important with the aerosols being produced with some of the drills, things like that. And again, droplets are larger than aerosols. They have a lot more virus, but they usually fall to the ground within 3 to 6 feet. So the largest effects at 1 meter, or there's a large effect at 1 meter, double the effect at 2 meters. And things like coughing, sneezing, singing, and shouting can all project viral particles further. Heavy breathing in gyms, choirs, crowded bars, singing, churches. That's why these have been some of the hot spots. And interestingly, I found a study that said talking produces just as many droplets as coughing. So, if you think, most of the time, if you're coughing and you're near someone, you're probably going to be polite and cough into your arm. If you're just sitting and talking, most of the time, and certainly in pre-COVID times, you didn't have a face mask on, you didn't have anything covered up. So I think this is a big reason why it's spreading so much at restaurants, small family gatherings, and dinner. People are sitting right across the table from each other, they're talking, and then those aerosols are coming out while they're talking. They're landing all over like the food in that area, and then you're breathing it in as well.
And if they're anything like my family, they're talking very loudly.
Then you get more people in more spaces. You get, more potential sources of infection, more people projecting it, talking louder, so on and so forth. Now, around this time last year, you may have heard, a couple people show up on the news saying, I think you need to leave your groceries outside for 3 days. The virus can live a long time on those surfaces. But it's really turning out that surface transmission is a minor, if not extremely rare route of transmission. So the CDC's guidelines at this point, it's that it's possible to pick it up via surfaces and then touching your mouth, nose, or eyes, but that's not the main way the virus spreads. That's because this surface transmission needs a very high infectious dose. So if you touch it on your fingers, you're only going to get some of the droplets. And you have enzymes on your fingers that can destroy the genome of the virus. You have oils that can disrupt the membrane. You have all sorts of antimicrobial particles and secretions in the mouth and in the nose. So it's going to be hard for those viral particles to get down deep into the lungs. It's a lot easier for them to get that way if you breathe. So you need a very high infectious dose to transmit it on surfaces. And as Dr. Thomson mentioned earlier, all the common disinfectants are very good at eliminating this virus. And also be careful in how some of these studies are interpreted. So there is one report that claimed, that the virus could be detected hours to days later on copper, cardboard, plastic, and steel. But one of these studies basically used the viral equivalent of 100 people coughing and sneezing in 1 square inch. So that's not really relevant to what we would see out in the wild. And also, you might have seen the reports about the virus was found weeks later on the Diamond Princess cruise ship, that one that had to be quarantined. Also, ask what they're looking for, because detectability does not equal viability. And also, because there might be remnants of the nucleic acid, there can be dead virus that you can still detect. Just because you can find it doesn't mean it's alive. It may have been dead for a few weeks and it's now no longer infectious, but you can still detect it. And also, just because you can find it doesn't mean that there is enough virus to cause an infection. So real-world studies that have detected viral A in the environment have found very low levels. Even fewer have been able to find viable virus after an extended period of time. And again, another way, identify and isolate infected individuals. We can do that through COVID testing. It's getting a lot better now. There's a couple different types of tests, and this chart from the FDA summarizes them nicely. So there's the molecular testing, which looks for the virus's nucleic acid. That's the PCR test, the RT-PCR test. Now, this is a highly accurate test. You can be very confident in the results if you take the test at the right time, but it takes a little longer for this test to run. There are the antigen tests. These are usually known as the rapid test, and they look for viral protein. Now, there's a high risk of false negatives with the antigen test, especially if you take it early after exposure. Now, if you take an antigen test and it comes back positive, you can be pretty sure that you can be almost certain that you do have the virus. But if you have a negative test and you have an exposure, it's probably best to follow that up with the PCR test as a backup. And also, these tests must be taken at an appropriate time. So say I went to, hang out with an unvaccinated friend, and I wasn't vaccinated either. And that person were to expose me to the virus. Next day they start coming down with symptoms. They test positive. Well, if I were to go get tested the next day, I'd come back negative, even if I was infected. The virus has an incubation period. So really you need to wait a good 4 to 5 days after the exposure before you can be very confident in the test results. Now, finally, there are the antibody tests. These cannot identify a person who's currently infected, but they could identify if a person was previously infected or if they had been vaccinated.
So here, what I want, this is an animation I made, best I got for animating things. But I made this for a test that we have for Streptococcus mutans, the bacteria that I work on for research. It's called a lateral flow assay. And so here, and this is similar to how the antigen tests work for SARS-CoV-2 detection. So you have a sample, a nasal swab that would contain the virus. So we'll pretend that these purple ones are the virus, not a gram-positive bacterium, and that there are antibodies that you get mixed with it with a detection color. And then it's going to pass through this cassette binding, and then there are antibodies that bind the virus as well, that are immobilized. And so then it will capture the virus and that detection antibody and make a line. And so you can see this really rapidly. And it's actually detecting the virus that's there. So I'm going to play this. So you put the sample in, and it's going to, by capillary action, pass down this thing. And so you get an idea here that, oh, there was things that bound and it changed color at the test line. And it's not going to look exactly like this, but they're basically these little cassettes that can show you within 5 to 15 minutes whether or not someone's positive. And it's a good measure of someone who would be infectious because it's actually measuring virus. I see that we have a couple questions, comments in the chat here. And I'm wondering, let's see. Yeah, there is a lot of issues with aerosol and not aerosol. And then again, like Dr. Fischer mentioned, what is aerosol versus what is droplet transmission? I think many people use them interchangeably. I know I did until March 2020, when I realized that there really was a distinction that needed to be made between the two. And based on what Alexandra said in the chat, I'm not sure I know what that's referring to in particular. So I'd have to look into that evidence.
All right. And so again, if person's walking around, they're sick before they even realize it. Again, this is why mask usage is so important. Just to reiterate, the mask needs to cover the mouth and nose to be effective because you can release aerosols through both and you can bring them in through both. So, if it's below your chin, below your nose, it's not doing you any good. Like N95s, KN95 masks, and the Korean equivalent, the KF94, offer the best protection. So you can now find some actual reputable KN95s on Amazon and other marketplaces. Mostly N95s are still going straight to healthcare professionals, as they should, but you can find some good KN95s or KF94s on Amazon and other places, and you can refer them. You can check with the FDA. They have like codes for which batches and manufacturers are reliable. But those two masks and also these combinations on the bottom, it's like the surgical mask with the tightly woven fabric mask or fabric masks with multiple layers and filters. Those have greater than 90% efficacy in collecting particles of similar size to respiratory droplets. So they bind them up, they dry them out, you don't breathe them in. Surgical masks are helpful. They're 60 to 70% effective against protecting others, 50% effective against protecting the wearers. But it's, it'd be a good idea maybe to combine those with a fabric mask as well.
But one is better than nothing.
Yes.
So everybody that's saying, well, now we got to wear 2 masks. You don't have to, but it's better. One, please, just one.
And before I was able to get my hands on the KN95s, I was doing the surgical mask, woven mask combination. And I actually found that held the surgical mask in place better for me, but I've got a big head, so.
I found something with like, I very early on, I put on I had a KN95 and I wore that out, back in April, and everybody was just staring at me. So the next day I went out and I had a cloth mask that had Pokemon on it that I got with my son, and nobody looked at me anymore. So it works for that too. It's more fashionable to have Pokemon than a KN95.
Yeah, and there's a lot of evidence out there now that the masks work. Germany has reduced transmission. The USS Theodore Roosevelt long-haul flights, the story with some Missouri hairstylists, all of them have shown that masks reduce transmission. And so, again, really avoid the 3 C's. So closed spaces with poor ventilation, crowded places, and close contact. So gathering in an indoor environment, 18.7 times more likely to get COVID than in an outdoor open environment. And more crowded the place is, the more people you've got that potentially would be vectors for disease. And then the closer you are, the more likely you are to breathe in that infected person's aerosols. So, what we've seen in, in some of the studies have shown this, and this is kind of bears out what I've seen doing contact tracing at the dental school. It doesn't really seem to be passing from casual contact or, going through the grocery store. People living together, riding in cars together, eating together, those small, close family gatherings. Some people were talking about, oh, well, had a barbecue. I thought it was safe to do that. Well, how much time did you spend inside in the air conditioning versus how much time did you spend outdoors by the grill? So, I mean, that's all very important things to keep in mind. And then people who dined at restaurants are twice as likely to develop COVID. And also, this is one point that we had trouble getting across early on in the pandemic. So when one of the first cases popped up after we reopened, someone was like, well, why wasn't an alert sent out to everybody? Well, that person did not have contact with very many people, and they're wearing PPE the whole time. So, I mean, if you have 6 feet distancing and you're wearing masks, being in the same building as an infected person is not necessarily a risk factor for COVID-19. Also, being in a large ventilated room with an infected person is not a risk factor for COVID-19. If you're somewhere that's, maybe in a large lecture hall or classroom, there's good air exchange, a person's 40 feet away from you, and you never got up close with them, you're at minimal risk there. And if you were in a huge building, if someone was in a different room and you never crossed their paths, that is not a risk factor. You don't need to shut down if things like that. I think we'll really just talk briefly about antivirals, since I think we're kind of running.
Yeah, I was
behind
thinking,
here.
so what I was thinking was monoclonal antibody therapy has been the biggest, I think, increase in the antiviral game. So there are a couple companies, Regeneron and Eli Lilly, that are making these cocktails of antibodies that do a really good job at pre-exposure or post-exposure prophylaxis. So if someone had an exposure that's at high risk, they get an injection subcutaneously. But you have to get it injected, and it has showed really good results in some clinical trials in preventing death and preventing clinical progression. And then another one that's pretty exciting is an oral drug, molnupiravir, that has shown pretty promising early studies in phase 2A, and now it's going into phase 3 studies. And so this is really important because remdesivir, which got a lot of publicity, is an IV drug. And so you can't take, it's acting against the virus, but once you're in the hospital, it's your immune system that's overreacting, that's putting you there. It's not really a viral induced destruction at that point. So having a direct acting antiviral like remdesivir is not necessarily going to be helpful, which I think is finally showing in a lot of the data as mentioned up here. But one that you could take orally, like you would with Tamiflu or something, and when you get influenza, very, very early on after you get that positive test, you take an oral drug that you can get from your pharmacy, take it for 5 days. That makes more sense. And I think it's showing in these trials now.
Should we move into vaccines here?
Hey, I just wanted to show another animation here on how, because this is important for vaccines too, because one of the things vaccines will do are generate what do we call neutralizing antibodies. And so what does a neutralizing antibody do? Is it binds to the virus and prevents it from getting into a cell and infecting a cell. And that's the main role of a neutralizing antibody. So here, what would happen normally is you have a virus and then you have a cell that it's going to infect. Those spike proteins bind the receptor and woohoo, it's infection time. And it can get into the cell and make new virus. However, when you have neutralizing antibodies like a monoclonal therapy or from vaccine or natural infection, they bind to those and neutralize the virus. It can't interact with the receptor and doesn't get into the cell. So that's kind of the idea for a neutralizing antibody. And then that one ran away. I don't know what I did there with that animation.
You can develop antibodies, you can develop an immune response by becoming naturally infected. The problem is with a disease like SARS-CoV-2 is that it kills a lot of people who get it. So or causes some of those long symptoms, or it's a very uncomfortable disease to have. So you want to be able to avoid having to do that, having to go through that process to generate immunity. So the best way to do that is vaccination. And I see Dr. Thompson's getting some high praise for his visual aids.
Thanks.
But the vaccination, it works to help make neutralizing antibodies, builds up immune memory, and builds up a T cell response, so you're armed and ready to go when you come into contact with the virus. And so if you were to ask a person, why do you vaccinate? I think a common answer would be to, maybe would be completely prevent infection. Well, yeah, that's the ideal goal, but it's not always possible. Our main goal is to prevent severe disease, to keep people out of the hospital and keep the healthcare system from becoming overwhelmed like it has a few times during this pandemic. And so it's looking like the Pfizer and Moderna vaccines are both very good at preventing infection entirely. And so that's great news, because that means a person who has the vaccine is exposed is probably not going to be able to pass it on to an unvaccinated person. All the available vaccines right now are great at preventing severe disease. And so they're all very effective at keeping people out of the hospital. And, if you think about the flu vaccine, if you hear the numbers in a given year, you might hear it's 40 to 60% effective. And that's effective against any signs and symptoms of disease. Now, a person who can be vaccinated and still get the flu. But if you look at the numbers, they're less likely to be hospitalized. If they're hospitalized, the stay is shorter and they're less likely to die. And so I think at this time last year, if you were told, hey, I can give you a shot that's going to knock this down into a common cold, I think anybody would have taken that. And right now we have 3 outstanding vaccines that, are very good at least 3, probably up to 4 now, that are very good at preventing disease, even mild disease, and are excellent, exceptional at protecting against serious disease, hospitalization, and almost universal against protecting against death. And so I'll start off with the Pfizer and Moderna vaccines, and I think Dr. Thompson will probably take over a little bit from the J&J. And are you going to talk about AstraZeneca? All right. So I'll talk about the mRNA vaccines that came out first. So the Pfizer and Moderna vaccines. What's the difference between the two? Oh, well, mostly it's the age guidelines, and they're looking to lower these age guidelines here. And I know more studies are going on in younger people. And I think this will be very important, because I think we are seeing spread in high schools and adolescents. But the Pfizer vaccine, 16 and up, given 21 days apart. Moderna, 18 and up, 28 days apart. I wouldn't get hung up on why 21 versus 28. It's probably just based on preliminary data and that's how they structured the clinical trials.
I think that's the main thing, is that they wanted to get these clinical trials done as fast as possible. And for the 1 month is why.
Yeah. And so, I mean, if you can't get it exactly at 21 or 28, don't worry, but try to keep it as close as possible. Both have a very similar mRNA platform. There's some small differences in the way they optimize the genetic sequence for the spike protein. And there are some differences in those lipid nanoparticles that they're put in. But they both have nearly identical efficacy after 2 doses, 95% against protecting against any disease, basically almost 100% protecting against serious disease and hospitalization. And I was fortunate enough to get the Moderna vaccine. So how does this work? So the vaccine is injected. It has these single-stranded RNA that codes for the spike gene for the virus. And so it's just a very small fraction of the virus's genome. The genome is over 30,000 base pairs. We just have the genetic material for this one protein. This one protein on its own cannot establish an infection, but it can build up an immune response that will protect you from infection. So the lipid nanoparticles stabilize the mRNA, and when it's injected, those lipid nanoparticles allow the RNA to get into your muscle cells near the site, near the injection site. After that, the RNA is then converted to protein in your cells, and you make the spike protein from the virus. The immune system then recognizes, oh, there's something weird going on in this cell. It's making a foreign protein. Then the body induces antibody responses, makes B cells, and makes killer T cells, and the person is now protected from disease.
So I want to, as we're getting here, I think that maybe we should just talk about the virus vector vaccines, and then we'll get into overall some of these slides. What do you think?
Go ahead.
So I think what we all know so far is how effective the mRNA vaccines are, and that they came out first because of the technology. The technology allows for faster production, as we'll talk about when we get to some new things. They went through rigorous testing, and they made it to market because of how much money was put into them. Clinical trials are expensive. Oh, okay, my kids are here. Yes. And let's see. There are adverse events that occur whenever you get a vaccine, and that's because your body is creating an immune reaction. It's not because you are being infected with these. But I know that.
And I want to comment on Jen Bowen's,
Yes.
what she put in the chat. Heard anecdotally that younger vaccine recipients have more side effects. Younger people tend to have more robust immune responses than older individuals. And so, they're probably getting a little bit more powerful inflammatory reaction. And then remember, a lot of the symptoms that you experience when you are sick are due to the immune response. So you produce inflammatory cytokines. Those can trigger the itching, the pain, and redness. They can trigger fever, fatigue, nausea. And also the immune response is draining of energy too. So, if a person's having a more powerful immune response, they're probably going to have more side effects. And though just because someone doesn't have side effects or like that doesn't mean that they aren't having an effective response to it. It just means their body isn't feeling it as much.
And I would say that it's not just anecdotally. There have been studies showing, like there's data on it too, that older individuals tend to have less reactogenicity to the vaccine, and younger individuals do. So here, just kind of showing Moderna and Pfizer first and second dose. The second dose tends to be much worse. There were some studies showing that if you've had a previous natural infection, your first dose might be worse, boosting that immune response. We did not have data necessarily during the clinical trials of getting the vaccine while you were pregnant. We know that it's advised to get the influenza vaccine during pregnancy because of how risky it is to get influenza while pregnant. And it's been proven over years because they're not actual infectious agents. They're just parts of a virus. And so the VAERS, or the Vaccine Adverse Events Reporting System, and the vSafe app have been collecting a lot of this data. And so this here is showing adverse events in pregnant women following the vaccine. There are 154 reports here. Miscarriage was the most frequent pregnancy-specific event reported to VAERS. Now, VAERS collects all adverse events. If you got hit by a truck the day after you got a vaccine, that would be a death that sometimes gets reported. I know that that's what happens in clinical trials, but VAERS is not designed to assess causality, but can rapidly detect safety signals. It's a really good reporting system. And so if we look at spontaneous abortion or miscarriage, 29 reports out of this pregnancy, there's data showing that that's around the same percentage as in a normal pregnant population. And so there were no real safety signals coming out of this so far with getting the vaccine during pregnancy, which makes sense to me as it is an inactivated vaccine, and we give inactivated vaccines to pregnant people, meaning they're not replication competent vaccines. One thing that I really wanted to make sure we touch on, this is the thing that I've been hearing the absolute most, but Francis Collins, the director of the NIH, this is from an article in the Washington Post. I'm worried, frankly, there are stories out there on the internet about how vaccination can lead to infertility. There's absolutely nothing to that. But when we look at people who are expressing hesitancy, in many instances, those are women of childbearing age. And I don't know if anybody else has had this, but this is something I hear more frequently as hesitancy, and this statement said it exactly. So why is this a thing? And so back in October, I believe, there's a former Pfizer scientist, left the company 9 years ago, is known to argue against vaccines, claimed the pandemic was over in the UK back in November, made a claim to halt vaccine trials, claiming that the spike protein resembles a protein involved in placental development, and that a reaction against the spike protein from these vaccines could lead to autoimmunity against this placental protein and lead into infertility. If this were true, then natural infection would also do the same thing because it's the same spike protein. So natural people would have fertility issues. However, there is no signal showing infertility from natural infection. Therefore, that logic, and that doesn't even go to mention the fact that the region he was referring to is 4 amino acids long. So spike is 1,273 amino acids, so the parts of the protein. There was 4 regions. Antibodies recognize much higher regions that are much larger than this specifically. From Dr. Langle, this is someone that Dr. Fischer went to, did his postdoc with and studies coronaviruses. Basically, the coronavirus spike and the placental protein in question have almost nothing in common, making the vaccine highly unlikely to trigger reaction to these delicate tissues. The 2 proteins share only a minuscule stretch of material. Mixing them up would be akin to mistaking a rhinoceros for a jaguar because they're wearing the same collar. And so the premise alone was incorrect, and in multiple ways, but just hearing that idea is enough for someone who's not necessarily at high risk of getting natural infection and having severe COVID. But the potential that there might be infertility, just being out there, plays a big role in people's decision making.
Yeah, and I mean, and if you don't get vaccinated, you're going to get COVID eventually. Unless we remain shut down and people stay inside forever. And so, that it's the same spike protein. It's that spike protein has that same antigen. So natural infection would have the exact same impact as being vaccinated. And plus, we don't know a lot of these long-term consequences of, COVID, and COVID can result in death, even in young, healthy, fertile people.
And we do know that there's new evidence that getting natural infection can cause inflammation that leads to affecting organs. There's some signals being reported on that it can lead to sexual dysfunction in males as well. That could lead to infertility. Margaret had a question: should people take greater precautions for the first 2 weeks after getting dose 1, 2, or both doses? So I would say, if you have not been vaccinated, the precautions that you're taking should be the same precautions that you're taking until 2 weeks after your second dose. If you get the J&J vaccine, I would hold out for that 1 month range. And then after that, I would still be maintaining those same precautions because not everybody is vaccinated. And I think setting a good example for anybody around you that might not have had the chance to get vaccinated is important, even though we know that there is some decreased risk in asymptomatic transmission.
Yeah, so you'll be very well protected 2 weeks after that second dose. And, there's going to be some relaxation in the guidelines. Vaccinated people, you can get together, if you're fully vaccinated 2 weeks out from the last dose, you can get together in a room without masks. But I mean, when you go out, you should still wear the mask because, give the poor service workers a break. I mean, you don't need to have them trying to figure out who's vaccinated, who's not. And I mean, and there's always variant strains and so on and so forth. I mean, you can have very high confidence in these vaccines, but, just continue to be cautious. You can relax a little bit, but don't be reckless, I would say.
And children can't get the vaccine yet. So that's also important. There's lots of people that they haven't done the trials for. For the viral vector vaccines, how do these work? I made this diagram. So basically we're using a virus vector. So in these cases, they're adenoviruses that have been modified to take out some of their genetic material that allows them to replicate in our cells. So they normally cause common colds in us, but they've changed them up. And so they can only replicate in insect cells and make new virus. So what they do is they grow these in insect cells in the lab that make a bunch of the virus, but then once they get into human cells, they can't replicate. But they've modified the genome of these adenovirus. So in this particular one, it's AD26 to incorporate the DNA gene of SARS-CoV-2 spike into that genome. So then this virus, which can get into our cells but not replicate, gets into the muscle cells at the injection and then into other cells, and is making the spike protein much like the mRNA vaccines. But it's not making any new adenovirus in there. So it's just another way to get that spike protein expressed inside the cell that resembles natural infection in just that one protein, because we know an immune response to the spike protein is so important. And so that's a viral vector. So the mRNA vaccines use this lipid nanoparticles to kind of sneak its way into a cell. This uses a normal viral entry mechanism from an adenovirus, but then the only thing that's happening is it's displaying the spike protein. And so we now have we're starting to get access to vaccination because this one came out. It's a single dose. February 27th is when it got its emergency use authorization. Had a huge phase 3 clinical trial. The vaccine's estimated to remain stable for 2 years at refrigeration. Nope, that is not refrigeration. That is like a normal freezer. And then in routine refrigeration can stay for 3 months. So it can go places. You can have vaccine clinics outside of places with minus 80 degrees Celsius freezers. Really good effectiveness at preventing moderate to severe critical COVID, secondary endpoints in severe and critical COVID. It's really high effective in these vaccinations. It's safe, it's effective at preventing symptomatic COVID. And now this is today, a press release from AstraZeneca that the US phase 3 trial has finally hit its primary efficacy endpoints. 76% against symptomatic COVID-19, 100% effective against severe and critical disease and hospitalization, 85% against symptomatic COVID-19. Novavax is on its way. This is just a protein subunit one. So basically you're making spike protein, assembling it into these little particles and throwing it at your cells saying, hey, come here and respond to me, basically. And it is showing really good effectiveness in its clinical trials in the UK and South Africa. We just have not finished our trials in the US. They do not, if anybody says I got COVID from the vaccine or don't come to my house, this happened to my family. My brother-in-law's dad was getting the vaccine and his sister said, we don't want, or you can't come to our house because you're going to be spreading COVID after the vaccine. That is not the case. It does not infect you. It is just a part. You can get COVID from something else right around the same time, which happened with me as well. My mother-in-law got the vaccine and symptoms of COVID on the same day from an unknown exposure somewhere. So that was exciting. And then this slide is really cool. So this is I was sent this on Twitter. I'm not on Twitter. I don't tweet, but apparently it's a great place for really good information with references. But just showing the different clinical trials from Moderna, Pfizer, Johnson & Johnson, and now AstraZeneca, I would expect FDA approval pretty soon. Emergency use authorization pretty soon from that as well, a couple weeks. Even though I think we just sold a bunch of our doses to Canada, but good, that's fine. Oh, thank you, Juliet. But just showing protection from COVID, severe disease, efficacy against mild COVID. These are all really, really good. And I think which one should you get? It's difficult to make direct comparisons because these clinical trials happened at different times with different levels of circulation of different variants and lots of different things that are just not directly comparable. And so they're all really effective in preventing mild disease, significantly reduce the chances of developing severe illness, and so far, almost complete protection against hospitalization and death in the clinical trials. And so which one should you get? The best one is the one you can get, and as soon as you can get it. And I think we can all just use this phrase now. Yes, absolutely. That's This Week in Virology. Every Friday, they have a clinical update with Dr. Daniel Griffin. He's been involved in a lot of the clinical trials, and he's an infectious disease doc in New York at Columbia University. Just awesome with all new updates. That's a great one. But yeah, little Hamilton reference there. They seem to be really good at preventing asymptomatic infection. We didn't know that at first, but real world data is coming in. Multiple studies in England, Israel, Mayo Clinic, showing where they're just sampling people that have been vaccinated, looking for asymptomatic infections, and they're not finding them currently. So I thought this was interesting. Once the Janssen and J&J vaccine got authorization February 27th, you see daily new COVID-19 vaccination doses and see how it just absolutely raised up because of the cold storage and how many they had through Operation Warp Speed. They had a lot of vaccine pre-made and ready to be rolled out. And then you see that sharp decline in cases from vaccination and natural infection, and not being right after the holidays. Maybe people were sick of their family after spending a bunch of time with them over the holidays, and so they weren't gathering as much. How are they performing in the real world? This is something that's really important because we have efficacy data from the clinical trials, but effectiveness data, once they're put out there, there's so many other factors. And so how are they doing? And so we have some great evaluation out of Israel. And basically, they're following up with the mRNA vaccines with the clinical trials. So looking at different intervals, 14 to 20 days after the first dose, and then once they get that second dose, looking at greater than 7 days after dose 2. And so 46% effective at preventing symptomatic illness after the first dose, after 2 weeks, once you generated your immune response. And then after 7 days, 92%. And that was any type of SARS-CoV-2 infection. Symptomatic, 94%, just like the clinical trials. 57% after the first dose, 2 weeks after the first dose. Hospitalization, real life, 74%, 87%. Severe COVID, 62%, 92%. So I advise everybody that first month before you get that second dose and 7 days after that second dose, pretend you never got vaccinated and hold yourself accordingly. You'll have some protection, but I wouldn't rely on that protection if I had the choice. Do I think children, so this is a weighted question. Do I think children that back at school at the beginning of March has had a significant effect on caseloads in Michigan? I don't think there is so much in-school transmission, but things associated with school, I think, is where it's being transmitted. Sporting events, gatherings, parties. I think that's where the transmission is happening. There's plenty of studies showing that if, like with quarantining students in school, I don't think it's happening there.
Yeah, and I'll say, and it seems to be spreading more at like the youth sports level, like high school sporting events, things like that. And I think some of that has to do with people in the stands yelling, getting close together, cheering, maybe pulling their masks down. I don't think there's much going on in the K-8 High school might be a little bit different, but also, I mean, at that same time, a lot of the restrictions were relaxed on bars and restaurants. So I think I've felt this myself, and I think it's like the pandemic fatigue has really hit a lot of people lately. I think people made it through the holidays. They kind of hunkered down for those bad winter months. Now that it's a little bit nicer, the restrictions are lifted a bit. People just want to get out and do things, and I think people have let their guard down a little bit. And the introduction of the B.1.1.7 variant is a factor too. I mean, it does seem to be more transmissible, but it doesn't seem to be doing, there doesn't seem to be terrible follow-ups to it, fortunately. So, I mean, I think there's a lot of factors that came into play there.
I think another big improvement, vaccines tend to be increasing long COVID, helping people with long COVID, and that there's a lot of anecdotal evidence out there that symptoms decrease after getting that vaccine. I know we're wrapping up. I put a ton of slides on here in hopes that we could provide them as well for your leisurely reading. Because there's a lot. There was one question about.
Can you hit slide 64 real quick?
I have no idea what the numbers are. Is that the DNA one?
No, it's the things to monitor with variants one.
Okay. But there was a question about the DNA alteration. No, I would not be concerned whatsoever about that. And that slide explains it.
Yeah, I mean, there's no way for, we don't have the machinery that makes DNA from RNA. So, we don't have the ability to do that. Then you'd have to get that DNA into the cell, into the nucleus. DNA doesn't go back through like that. And then also, you'd have to find a way to integrate that DNA into the genome, and we don't have that. So you might ask about, well, what about someone with HIV? They got a virus that reverse transcriptase and integrase. Well, that just doesn't pick up RNA and DNA willy-nilly. It's very targeted towards the HIV genome. So there's just nothing in our biological knowledge that indicates that the RNA vaccine could cause any alteration to a cell or to the genome of a cell, and it's certainly not going to get anywhere near the reproductive cells, so it wouldn't cause any defects to be passed on. Well, one thing with the variants that's been popping up, there's a few headlines that showed up saying, oh, Pfizer and Moderna's shots were 10 times less effective against a particular variant or not. And okay, how do you define effective? Well, I define effective as, does it protect against disease? You can also look at how well do antibodies bind. So they may bind the original strain really tightly at a level up here. They may only bind the variant here. Okay, so maybe that's 10-fold less, but if your protection threshold is way down here, that level is still going to do you a lot of good and protect you from diseases. So, I mean, be very careful about how people are, reading too much into headlines, and because I think, there's a lousy job of scientific communication out there, and people run with things that might not be completely accurate. Yeah, I do not want to be part of a medical experiment, and technically this all is a big medical experiment.
Real time.
Well, I'm thrilled to be a part of it.
Yeah.
You're going to be a medical experiment too if you catch the virus, in the wild.
We had a question about what are the most effective ways to educate individuals on public vaccine safety? I'm not positive. I'm working on it. But one thing is that people relate and they remember stories and anecdotes, real life experiences, stories, anecdotes, and not necessarily the facts, not numbers, not percentages. And that's my problem because I live on those. And one thing I've been messing with, on Facebook, I've been putting stuff out. Nobody looks at my Facebook ever. And then all of a sudden I put this out there about the vaccines. And within 5 minutes of going outside after hitting post, my wife says, hey, you might want to get back here. You have 15 questions on your post. And it got shared over 100 times. And I had people sending me messages thanking me because they needed someone they could trust. I'm like, well, first of all, why are you trusting me over anybody else? but that they knew me. They felt comfortable listening to me. They grew up with me. They knew my wife. They knew my brother. And I knew somebody that shared it. And so I think that's something to go from. And when to come to an end? I don't know.
Great question. You know, I mean, I'm very optimistic with the vaccine rollout. I mean, for the emergency use authorization, they were looking at 50% effective against the disease as being a threshold. I mean, we got stuff 75%, 95%. I mean, that's just incredible. And it's just a really remarkable breakthrough. And I'm really loving the data that's coming in, showing that a lot of these vaccines can protect against asymptomatic infection. I'm loving getting all the texts from my friends saying, "Hey, I finally got in. I'm going to get dose 1 today or dose 2 today."
Not giving up my shot.
Yeah, and so I think we're really headed in the right direction now. We were stagnant and if not a train wreck for a lot of 2020, but I think I'm very optimistic about things. I think we'll be dealing with masks for a while now. I would expect masks probably until this time next year, maybe, but in public. But I think we can get back to maybe some of the more private gatherings, groups of vaccinated people getting together. And then I'm very optimistic for this summer. So, just encourage all your friends and family to go get protected. And, maybe we can see something resembling whatever the new normal is going to look like.
And just
in the
one
next
last
several
word
months.
on the variants. One awesome thing about these the mRNA platform is how easy it is to manipulate and change on the fly. Moderna just sent out a press release a couple days ago, one day ago, that they have the B351 variant. So the one that evades a bit of neutralizing antibody. They've already sent it out to the EU to start testing it in trials. And Moderna is starting a trial in children 6 months to 18 years of age. I'm trying to figure out how to get my kids involved in that. They will not be happy with me to get a shot, but yeah, I know we're out of time and overtime, and I'm happy to stick around and answer questions by email. I think everybody had do you have our emails? Please reach out.
Yeah, and then we can send I guess we could send the slides maybe to Jill to distribute that.
Yes, if you could send them to me, that would be great. And do you have a problem with me providing, I had originally said we'd provide the recording on the website behind the password, but if you're okay with providing the recording to all attendees or others that ask, that's
Please.
up to you.
Please, send it far and wide.
Okay.
Yeah, we debate about all that stuff, and it's just important. I feel like we can reach people doing things like this in some way. I know I talk forever and I'm long-winded and I'm long written on all my posts, but some people are reading it still. People are dying for information they feel like they can trust.
There's just so much out there. It's hard to sift through everything.
I've spent 12 months doing it.
Well, for some of us too, some of the microbiology has been 20 years past. And so it would be nice to be able to go through the slides and take my time and relearn some of that stuff. So were there any final questions, or feel free to unmute yourself if you don't want to talk? I mean, if you don't want to type. Okay, thank you so much, both of you. We really appreciate it. I've heard your talk. This will be 3 times now, and I learn something new every time I hear it. So
lucky you.
I love this. I find this fascinating. So thank you. I
Thanks
really
for having us.
Yeah, thank you so much for the invitation and everybody's attention. And your nice comments as well. Henry says hello.
Hi.
And so does Lena. He wants me to be done with my meeting.
That's it, and I appreciate your attendance, and we will get the presentation and slides out to, if you want them, send me an email and I will email them to you. MDMLG people, we will put it up behind the password protected part of the website. So thank you. I appreciate your attendance, and we will see you in May, I guess.
Thanks again, everybody.
Thank you.